Hello, my name is Yu Qing. I'm leading the ARI research program on resistance gene discovery. And the future challenges of weed management are posed by the evolution of non-target-based, especially metabolism-based resistance to herbicide of different modes of actions. So this program is to provide industry with new knowledge in novel biochemical and genetics of herbicide resistance in Australian major crop weeds by focusing on identifying genes and their functions involved in non-targeted resistance to prey and emergent herbicides. So in the next few years of ARI Phase 6, research priorities are given to first, cytochrome P450-based multiple resistance. P450-based cross-resistance to multiple herbicides is widespread in annual ryegrass in Australia and the world. Our recent work has demonstrated that a single P450 gene can endow resistance to herbicide of at least 5 modes of action with unpredictable resistance spectra. And our most recent work has identified that this resistance even extends to existing and new pre-emergent herbicides. So the further work is to identify more herbicides that might be made redundant by this major P450 gene and identify more P450 genes that are involved in cross-resistance. Another research priority is non-target-based resistance to the 3 non-selective herbicides, glyphosate, glufosinate, and paraquat. Non-target-based resistance to these 3 herbicides either very common or emergent in a number of grass weed species, such as annual ryegrass, Johnson grass, banyan grass, and goosegrass. Our recent work with glyphosate-resistant banyan grass discovered that an enzyme that can metabolize glyphosate and a membrane transporter that can move glyphosate out of plant cells, and thus reducing tissue glyphosate concentration and endowing glyphosate resistance. So in the next few years, we will endeavor to uncover non-target-based resistance to glyphosate in annual ryegrass and the Johnson grass. Of course, and high-quality genomic sequencing, genomic information is a great help to this challenging task. And genomic sequencing for annual ryegrass is currently underway by the effort of an international weeds genomic consortium. We are planning to sequence Johnson grass genome with Chinese collaborators. In collaboration with Chinese scientists, we have recently completed a high-quality genomic sequencing for goosegrass, and these genomic resources will be used for gene discovery for non-target resistance to glufosinate and paraquat. So all this genomic-based resistance gene discovery will help or influence the best weed management strategies, such as how to judiciously select herbicide mixture and rotations, taking consideration of P450-based cross-resistance, and help in the future genetic approaches to reverse resistance, such as RNA technology and genomic editing. And all this work will ensure ARI and Australia at the forefront of international plant science research.